Hello and welcome to today's webinar. My name is Paul and I'll be in the background answering any WebEx technical questions. We have a few housekeeping items before we get started. If you experience any technical difficulties during the WebEx session, please dial 1-866-779-3239 or you can message me, the WebEx producer, by using the Q&A panel. During the presentation, all participants will remain in listen-only mode, and as a reminder, this event is being recorded for rebroadcast. We will be holding a Q&A session at the conclusion of today's presentation. We encourage you to submit written questions at any time during the presentation using the Q&A panel at the bottom right of your screen. Please type your questions into the text field and hit send. Please keep the drop-down as all panelists. And with that, I'll now hand it over to Adam McChesney. Adam, you have the floor.
Thank you, Paul. And thank you for those joining both near and far. This is the first part of a four-part series discussing the Stealth Autoguide, Medtronic's cranial robotic guidance platform. In this series, we will cover the initial clinical experience of 3 neurosurgeons across the United States since market approval in November of last year. Today, the discussion will be focused on the development from inception through preclinical trials and the value of technology integration, robotics with stealth navigation, Midas Rex, and even O-arm imaging, into a workflow you're likely already familiar with. I'm pleased to introduce Professor Stefan Wolfsberger of the Medical University of Vienna. Professor Wolfsberger has been instrumental in the development of the Stealth Autoguide and has presented at national meetings to standing room only audiences on this topic. Before we get started, first I want to additionally welcome our panel made up of medical education and marketing colleagues from several regions across the globe. As Paul said, we will, as a panel, collectively be moderating questions submitted through the application at the lower right-hand side of your screen. We will also reserve the last 10 to 15 minutes for open dialogue in which you can voice your questions by raising your hand. Additionally, within the application at the lower right-hand side of the screen. I thank you for attending and again have the privilege of introducing Professor Stefan Wolfsberger. Professor Wolfsberger, the virtual stage is yours.
So thank you very much. Thanks a lot for inviting me. I hope everybody can hear me. Correct, Adam? Okay.
Yeah.
Thank you. So my name is Stefan Wolfsberger. I'm a neurosurgeon consultant at the Department of Neurosurgery of the Medical University of Vienna in Austria. And I'm really proud and excited to tell you about our last 7-year project developing a small, miniaturized robotic device for neurosurgery, which finally was called AutoguideTM. And on this first slide, you can see the final version of AutoguideTM here, and me taking a biopsy in a mock OR. And I would like to show you today how everything started. So I have to disclaim that this study was sponsored by Medtronic, and I'm an advisory board and educational consultant of the company. But even more, I'm biased because this robot was actually originally developed in Austria, in my home country, which creates, again, I think, more bias than this disclaimer. So to start off with, I would like to tell you a bit about our motivation. So why did we want to develop a robot? The thing is, stereotactic neurosurgical procedures, in my opinion, are getting more common. They're minimally invasive, they're super efficient because they target a specific neural structure. And we think that especially with molecular pathology, we are doing more and more, especially biopsies, because our pathologists want molecular pathology of every recurrent tumor, because they want to specifically target these tissues. So they're getting more and more common, and we think they should be fast and easy to set up. They shouldn't add significantly to OR time, and therefore seamlessly integrate into the existing workflow. So let's have a look a bit what kind of stereotactic systems we have. And there is, as you can see, the stereotactic frame. You all know this, and this, if we want to be super highly accurate, we still may use this stereotactic frame, but we know it's not a fast application, not a fast setup. What about frameless stereotaxy? I think this is mostly used nowadays for the regular biopsy, but we all know it may not be super accurate, and some of its adjustments are very cumbersome. And there are already the robotic devices on the market. You can see here the Renishaw robot. But we know most of the robots are very big and bulky, and well, they add, in fact, to OR time. So we think that there's a critical need for a new stereotactic device for neurosurgery. And this is what we found, we thought we found in this very small robot, again, made in Austria, originally called ISIS-1. And it was designed as a modular guidance system for, in fact, surgical tools. And primarily it was marketed for interventional radiology, where it's also CE marked, so that in fact the radiologist doesn't have to put his hands into the X-ray area. And what it can do, it can align a surgical instrument with a predefined trajectory. You can see these two plates that move against each other, and you can see these two manipulators or end effectors that hold an instrument here and also move against each other. And with this, it can actually target an area of 4 times 4 centimeters and angulate in an area of 30 degrees, plus minus 30 degrees. And in fact, we thought that this may be actually enough for most of neurosurgical targets. And the idea was hook it up to a standard navigation system, get the input from the current position of your instrument, and let the robot then do the fine positioning of your, for example, biopsy needle. So you see here was our initial comparison. Most relevant robots on the market were the Neuromate and the ROSA. You all know these systems. And here is this what we call surgical trajectory platform. And I think actually the scale is quite realistic. You see how small it is compared to the other robots and how much it actually fits into your normal surgical procedure. Here again is a comparison. You see the weight. The AutoGuide is weighing only 1.5 kilograms compared to more than 200 kilograms of the big robots, and its dimension is very small, as you can see, 25 times 10 centimeters approximately, whereas the other ones are in the meter range. So here you can see our actually way how to develop a robotic system for neurosurgery. So it was quite difficult, especially at the beginning, for the technical development to have an interface between robot and navigation and to get this system through our governmental agency. So, but first of all, because before we embarked on the certification to commence this study, we had to do a preclinical trial. And here you see the first preclinical trial. Here's a skull base fitted with 9 titanium screws, as you can see here. Here's the very first version of the ISIS-1 robot equipped with a pointing device. And we were doing targets. We were doing, in fact, biopsies either with ISIS-1 or with a manual biopsy arm that you all know, so the vertex arm. And we had 9 neurosurgeons targeting 9 biopsies. And some of them were very experienced stereotactic neurosurgeons, and some of them had never done a biopsy before. And it's very interesting what we could see as results. So using the standard biopsy arm, it took us mean 3.7 minutes to do 1 biopsy. And with the robot, it took 2.6 minutes, which was significantly shorter. And coming to the error, target error, the standard biopsy arm was 1.2 millimeter inaccurate, whereas the robotic guidance was only 0.6 millimeters accurate. And again, this was significantly more accurate. So significantly more fast and more accurate actually led us to believe that this could be a good idea. So next we had to develop robot attachments. First for biopsy, you see a drill guide, a very basic drill guide. Here needle holder for the standard biopsy needle. And here for catheter placements, we needed to develop something for electromagnetic navigation. Yeah, and you see all these metal-free, non-ferromagnetic attachments for the drill, and here for the catheter with the EM stylet inside. So then here you see our basement at the department with some cadaver tests. Here you see the ISS-1 again, the first version we had, very first version with 2 arms. We were very scared this robot may move, may not be stable enough. So at first we used 2 arms. We tested the draping. We tested biopsy. We tested shunt draping, as you can see here. Here's now the peak version, so the non-ferromagnetic version. And actually, some of our guys really had to dive into. Here you see our PhD student, Dr. Minchev, who is underneath the drape in the Mayfield clamp, testing the shunt drape. So then here again, you see the shunt positioning during the test. And then we went to our ethics committee and our governmental agency and asked for approval of this clinical research project. And in 2013, we were very proud that after some negotiations, it was possible to do a study, which we called application of the robot in stereotactic neurosurgical procedures. And it was approved for biopsy, catheter placements, and depth electrodes for sEEG. And you see, until now, in 7 years, we have performed 150 cases with the robot. You see our patients. Most of them were tumor biopsies. We did some depth electrode placements, more than 100 electrodes by now. We did some shunt placements at the beginning and some abscess drainages, but only at the very beginning. I don't want to talk too much about this crowded slide, but you can see that we did bilateral, we did prone position, and we did a lot of different histological diagnosis biopsies. Also concerning the target volume, very different lesions, very small lesions were targeted, and of course also very big ones. And here you see the error, which is interestingly, so here the real error on post-operative imaging. We developed a very special method of calculating this error so that it is actually accurate. And you see at the entry point, so that's the CT drill hole, 1.5 millimeters mean error, and at the target 1.6 millimeters mean error. So it's very, very accurate. And coming to the time, to the OR time, of course, there's always the question, does it need more setup time? Does the surgical time increase? Well, we're not sure. Yeah. So you see the setup time was around 12 minutes and the surgical time about 19 minutes. And this is overall, of course, 1 biopsy or 1 depth electrode. So it's quite fast, we believe. Yes, it's sEEG, but I will come for this, to this subject a bit later. So let's start with our clinical research project. First of all, we wanted to test this robot in our very, very standard Burr hole procedures. So a standard biopsy procedure. And honestly, I can recommend, if you want to try to use this robot, the first thing you should do is a typical frontal standard Burr hole biopsy. So this is our OR setup for optical tracking. So the robot is mounted to the Mayfield clamp. There's a special adapter here, and what is called multifunctional arm, this multi-joint arm, where it is fixated, and the robot is then brought into its working space and moves to the final target by itself. The same is possible for electromagnetic tracking. We also had this, as I told you, special non-ferromagnetic attachment for this catheter stylet assembly. So here you see the OR that was probably before our first case. Yeah, so here you see the adapter for the Mayfield clamp with the different starburst adapters. Here is 1 arm now. Yeah, the robot itself has also different adapters so that you can mount it either on either side, to the front, back, and on the left or on the right, just as you wish and as you are familiar with. So then this is during OR. I apologize for not covering my nose in Corona times. So But you can see the robot is very small. The surgeon's position is just at the same position as normal, so you don't have to change your position at all, like with the big robots. And if you don't like the robot anymore, if you don't need it anymore, you put it just to the side. You open the arm, put it to the side, and you can continue your regular surgery. So, of course, we have to look a bit at our steps of the procedure. So here you can see our standard burr hole procedure. Now how we do, we localize our entry point with a pointer, like we always did. Then we do a skin incision and cauterization, just as usual. And then this is different. Before we pre-positioned the vertex arm, now we pre-position the robot. But now we don't have to align the vertex arm, but the robot does it automatically. Then we drill the bone. We can do that either through the guide tube or outside the guide tube, just as a regular drill hole. And then we insert the instrument through the robot's guidance tube. And this can be a biopsy needle, catheter, depth electrode, whatever you wish. So here is the most exciting case we had, because it was our case number 1. As I told you, a stereotactic frontal biopsy. And I hope you can see it all kind of smooth. So you see, we were very, very careful in this case. So a left frontal biopsy, as you can see, the robot was positioned with 2 multifunctional arms. Yeah, because again, we were really scared. There may be any relative movement. Shaving and draping was just as normal. And draping of the robot is a bit like an endoscope. You put a bag over the robot, you put its 2 sterile manipulators in the holes of the end effectors, and then have the guided tube again. Look, a bit like the endoscope. And then we checked 100 times our entry point. Yeah, just to make sure you don't see a drop of blood, because we're again very accurately doing hemostasis. And then you bring the robot in when you have done the skin incision. You bring the robot into place when it tells you position OK, and it did that at this huge computer that was attached to it. Then it could register. Registration meant that the robot is registered with the navigation system. And then finally it moves to target. And you can see it moves to target very accurately. It moves actually to target always to 0.1 or 0.0 millimeters, yeah, inherently. Yeah, so, and that's within, let's say, seconds. And when you're there, you just do your biopsy. You see that here, just your regular biopsy. And our first biopsy was taken from the contrast enhancing area of this probable glioma. So we took our biopsy in a standard fashion. We always check our specimen with 5 ALA fluorescence, and we were a bit surprised. It was negative. Still, it was inside the contrast enhancement. So because it was so easy, already during the first biopsy, we changed the robot position to a second trajectory through the same burr hole, again 0.1 accurate. You see, it's inside the PET hotspot, and we took a sample at the PET hotspot through the same burr hole, again put it under the blue light, and you can see there's a very small focal ALA positivity, and it was judged a grade 3 glioma in the end. So this was our first biopsy. And then after 25 procedures, 17 shunt, as you can biopsy, 5 shunt, and 3 cyst drainages, we published this paper in Journal of Neurosurgery. And we just wanted to mention that this robot can significantly improve accuracy, we believe, compared to a frameless stereotaxy, and reduce instrument positioning by time, because we realized that the positioning is so fast using the robot compared to the vertex arm, where it's quite cumbersome to do a manual instrument position. And you see here our accuracy, and of course we were highly careful, but at the entrance 1.3 millimeters, at the target 0.9 millimeters, and using not only scalp screw registration but also surface registration, we believe this is quite accurate for a regular frameless stereotactic procedure. So, and then we embarked on the depth electrodes for sEEG with the robot by a minimal invasive drill hole procedure. So first of all, we had to go through a preclinical phantom study again. So you can see here, it was a very short study. We did 5 electrodes with manual setting and 5 electrodes with a robot in a skull, human skull. And you see again these differences in accuracy and in duration, and I think this was enough for us and the ethics committee that we were allowed to use the robot for this study. So again, I would like to introduce you to this twist drill procedure. The twist drill procedure was something that my colleague Dr. Dorfer invented, which is very minimally invasive, and you will see it's a stab incision procedure. What you do is you position a robot first with a distance guide. You do a stab incision through the robot, can enlarge it a bit and cauterize as needed, and then you insert a guidance tube. So you lock the guidance tube to the bone with a K-wire. And then you measure the drill length and perform a twist drill hole of approximately 3 millimeters. And through this twist drill hole, you insert the instrument, in this case the sEEG bolt that will guide your depth electrode to the target. So what does this look like? So again, this is the invention. This is the special guidance tube. You see it has sharp teeth at the end, yeah, which locks to the bone. It has a window here on the side so that you can see the instrument inside, and it irrigates out the drill burrs. And here you see the K-wire inside. And with the K-wire, you center punch and lock this to the bone. So that's what it looks like. So first of all, when the robot has moved to target, you introduce the K-wire and lock it to the bone, so that this position doesn't change anymore. And then you slide the guidance tube over the K-wire, so that the K-wire also, so that the guidance tube also locks to the bone at the perfect accurate position. So then you measure the drill length. This was the drill we had actually during the study. You do stereotactic drilling through the guidance tube, and then you insert the bolt and the electrode, as you can see here. And here's a postoperative verification that we always did. You see, for example, this is a very small sliding of the electrode, but of course, because if there are different layers of tissue, you will always have a small sliding of the, in fact, very flexible electrode. But the position was very, very accurate. So here you see a live video that's actually from our publication in Journal of Neurosurgery. So the robot is moving to the first target again. As you can see, again, 0.1 millimeter accuracy inherently in the system. So what we then do is our small stab incision. We open the stab incision to some coagulation, and then we drop in the guidance tube. And at that time, we used quite a big hammer for our spine colleagues, as you see. We locked the guide tube to the bone. At that time, we did a center punch for the drilling. Now we do it a bit differently. We first center punch and then lock the guide tube to the bone. We believe this is more accurate. Then we do adjustment of the drill stop device, yeah, because our guidance tube is exactly 10 centimeters long. We do monopolar coagulation of the dura, yeah, of the dura and pia actually, to be able to prevent any subdural bleed. Then we drop the bolt, as you can see here, yeah, and that's the screwing of the bolt. Yeah, we know every round of screwing makes half a millimeter. Here we remove it, and then we calculate the distance of the depth electrode. We set the depth of the electrode and enter it into the bolt. And actually, if the next target is close enough, the robot can just move to the next, to its next trajectory, as you can see here. Yeah, what it will do if we have a few electrodes in a very short position. Okay, here you see the results of this depth electrode implantation study, and again, this was very accurate. But I have to admit, all of them were done with a skull screw registration, which by definition is of course highly accurate. Yeah. So this was the publication you can read in 2017 in Journal Neurosurgery, and we concluded that it increased the accuracy of the manual technique, which was also done with the same instrument set, by 60% at the entry point and over 30% at the target. And in fact, what it also did, it increased speed of the procedure to around 10, not maybe even less 10 minutes per electrode. So then we thought this minimally invasive set is a very interesting procedure, maybe also for biopsies, because it seems to improve accuracy and reduce invasiveness. So we thought, let's adapt this instrument set for biopsy. So you can see here we did some adaptations. Not only is the hammer smaller, we also had some continuous tracking of the robot here, because we wanted to know where is the where do we accumulate inaccuracy, or where are the areas of highest accuracy. So we wanted to know what happens during a biopsy. And as we all know from your frameless biopsies, once you lock your trajectory, you don't know what actually happens to your accuracy. So here is a graph showing the procedural system stability. You see one line is about half millimeter. Here's 1 millimeter inaccuracy. Here's 2 millimeter inaccuracy. And you see here we started our procedure. Here we placed the K-wire drill guide. Here we started drilling, and of course you can imagine there's a lot of drag on the drill, so we had some inaccuracy, but this reverted again to about 0.5 when we stopped drilling. And here we placed our biopsy needle with about 0.25 millimeters. So with this, we thought we can actually use the stereotactic minimally invasive instrument set also for biopsy. You see again, stereotactic drilling and then entry of the biopsy needle through that guidance tube. We needed a bit of a bigger burr hole, about 3.2 millimeter drill hole. Okay, let's have a look at the case. So you see a case of a 53-year-old male with a right-sided hemiparesis, and he had this left subinsular posterior internal capsule lesion. You can see it actually here. So the robot already moved to the target, and you can see this is real time, and we are again at 0.1 millimeter or even 0.0 millimeter accuracy. So we lock the target trajectory, and then here we have a continuous update of our current accuracy that we could see. You can see it on the video. So we have locked it, the guide to the bone. We are now entering our K-wire, and we are now locking the K-wire to the bone, measuring our drill, and then do a stereotactic drilling of a 3.2 millimeter drill hole. Okay, we're clearing our burrs, and then we're entering the biopsy needle. And you see, it's a very deep target. So we're coming actually prone position even. Yeah, so we're coming from posterior, from parietal. We're entering the lesion with a tip stop 21 centimeters. So it's actually exactly what we can reach with this type of biopsy needle. And here we're now doing our getting our results. You see, oh, sorry, I wanted to show. Yeah. No, sorry, I think, yeah. So I wanted to show we also always did a check, accuracy check on the postoperative imaging. So what you can see here is the preoperative trajectory, so the planned trajectory in yellow, and the real trajectory in red. This is calculated from the center of the drill hole, and you see how small the drill hole is, yeah, and the center of the area where we took the biopsy, yeah, and you see the error at the entry point 1.5, error at the target point 1.6 millimeter, also 21 centimeter depth. So we thought that was actually very good. Another case, 65-year-old male with a limbic glioma, diencephalic glioma, and actually many centers refused to do a biopsy there. It was very close actually to the PCOM perforators here, yeah, at the posterior perforated substance. So we did a biopsy, and you can see how accurately actually we reached the target, and it was an oligodendroglioma grade 3. So what is interesting, it doesn't depend with this instrument set where you take the biopsy. So for example, temporal biopsies or posterior fossa biopsies usually have very thick layers of muscle on your way to the outer table of the skull, and usually you have to do quite large skin incision. But see how small the skin incision is with a minimal invasive instrument set, because it doesn't make any difference if you have a thick skin galea layer or a thin one. Yeah, so it's always about 12 millimeter skin incision that you need. So here's our publication about the minimally invasive instrument set for brain tumor biopsies. So what we concluded is that we significantly improved accuracy and reduced operating time, because operating time, again, is always the same, no matter if it's temporal or posterior fossa or simple frontal convexity biopsy. It's always the same. The instrument set and the procedure is always the same. We were very proud that this was actually published on the cover page of Journal of Neurosurgery in January of this year, as you can see. So and then what we want to check is what's the difference between the regular use of a robot in the clinical routine and the regular use of the vertex arm in clinical routine. Because at parallel, we had different people using vertex arm, and we had different people using the robot. And this is what was just published in Operative Neurosurgery. You can see that, and I think this is very interesting to see. So here you see, sorry, this is in German, the manual accuracy, the accuracy of the manual biopsy at the entry point and at the target point. So every line is 1 millimeter. And you can see there is quite some inaccuracy. Yeah? So the green circle is around 1 millimeter. And now here, look at the robot-guided biopsy. At the entry point, it's always like system inherent, it's always more accurate. You see many of those dots are within 1 millimeter accuracy, but even here at the target, you never exceed 3 millimeter inaccuracy. So we thought that compared to the vertex arm, so compared to a manual adjustment, we can significantly improve accuracy without increasing operating time. Because what I would really like to mention is, of course, using vertex arm or any other manual biopsy set, you can of course reach 0.0 millimeter trajectory alignment, but it takes you quite a long time and it can be quite cumbersome. Whereas the robot moves to this accuracy in seconds. So in the end, we had to advance the system to, let's say, a commercially available system and to put in all our experience. And the system was in the end called AutoGuide after many different names. And you can see there were lots of design studies. This is just one I would like to show. But what we knew we wanted to do is a continuous tracking of the robot and of the needle, so that you're never blinded to your accuracy when you lock it. Yeah? And of course, we only wanted to have one arm, yeah, with a special locking technique that we also developed for it. So here you see the final robot AutoGuide in a mock OR. You see the big computer has vanished. It's now all included in a remote control that can be put on the nurse's table, and the robot itself. Here's one arm. Here's this ratchet that allows opening and closing of the system underneath the drape, the surgical drapes. And here is the reinforced end effectors of the robot. Here you can see it. Yeah? And the continuous tracking. So let's have a bit of a closer look. So how would you do a biopsy now? First, you would pre-position the robot manually, yeah, within its working space of 4 times 4 centimeters, and it will show you when it's outside of reach and inside of reach. So here the video, then when you are inside of reach, you can actually press a button and the robot will move to target. It doesn't need to register anymore, it will just move to target. And again, here you can see these reinforced guidance tubes, the reinforced end effectors compared to the previous version. So again, then you would fix the K-wire to the bone first. We realized that punching a center pivot first locks the accuracy, and then you slide the guidance tube over that K-wire. You see that here, how it is done. Then with Midas Rex, we developed a very special drill attachment, and you can see here, this is an adjustable drill stop, so you can just dial in the depth of the bone. So what you do is you measure the thickness of the bone on your imaging, and you just dial 3 to 14 millimeters, which covers about more than 90% of at least Austrian skulls. And then you know that the drill will not enter deeper than this depth. And in fact, you don't have to set any depth stop anymore. Here you can see what the drill looks like. You use the Midas Rex drill with this special attachment, and there are special types of drill bits with Burr less holes, rectangular holes, that will finally allow the needle or the depth electrode to slide in through a very, very small drill hole. Then you perform the needle biopsy just as usual. That's my initial slide actually. So coming to the future, I would like to share with you some future ideas and applications. So what we are already doing is a study on high precision pre-resection tumor sampling. We have a 7 Tesla MRI in Vienna, and they were very interested in taking biopsy samples from their spectroscopy hotspots, which are many more hotspots than you can get from 1.5 or 3 Tesla. And so the idea is prevent a brain shift completely, and when you have exposed the bone, take the robot, take 3-4 samples from predefined hotspots and send the samples to your pathologist and correlate them with your MRI scan. What about laser ablative procedures? This is already working. In fact, you can use the robot for Visualase laser ablation procedures. There's an instrument set included. What would be interesting for the future, maybe a set drive that could also advance, of course, the laser fiber. Yeah? This is a project that is already running, yeah, to use the robot as an endoscope holder and guidance, because of course, because it has a joystick included, you could use the joystick to move the robot within a certain safety zone. And for example, when you're talking about pituitary endoscopy, skull base endoscopy, then it is very cumbersome first using your assistant for many hours of surgery, holding the endoscope. And on the other hand, the holding devices on the market are either super expensive if they are very stable and easy to move, or they're not stable, especially if you lock them, they're swinging after, yeah, or they're very bulky. So I think there's room for improvement, and the robot, if you have one, could be an ideal instrument to hold your endoscope and to use the joystick to move it within a certain area. And of course, for ventricular endoscopy, same thing. If you have a safety zone of, I don't know, 0.5 centimeters or 10 millimeters, you can very, very precisely move inside your ventricle without creating any damage. We have been looking at the brain trocar, so that the robot guides the brain trocar to target and then allows for small movements. So this is also on the radar. We always asked about DBS, so if we can use the robot for DBS. Well, of course you could, but on the other hand, we have to compete against Lexel frame typically, so a super high accuracy. So we have to probably adjust our workflow, probably to a non-navigated robotic DBS, so using the robot internal measurement systems for positioning. And then there was the idea of doing trigeminal rhizotomy, so thermocoagulation, balloon compression with axial needle guidance guided by the robot. So just to show you some ideas, so here's the Endoscope-Tracker Assembly. Here's you see the S-Collab endoscope and the electronic SureTrack system, how you can guide the endoscope. Of course, you could theoretically introduce it into the guidance sheath of the robot here. And interestingly, my computer doesn't move. Okay, sorry for that. So here are the joystick movements of the robot. You see me here with the remote control moving the endoscope, and the endoscope is guided via navigation by optic tracking here. So this is just a mock OR, a proof of concept that it works, of course. So to conclude, we think due to the miniaturized design of this robot, it was very easy to integrate it into our standard surgeries, such as biopsy, such as shunt placement if you want to do it, and such as depth electrode placement. And if you don't need the robot and do as a resection thereafter, you can just put it on the side. It will not interfere with any of your normal surgical procedure. And we think that what we could show in our preliminary clinical data, it can significantly improve the procedural accuracy, because it always has an accuracy of 0.0 or 0.1 millimeter, and it reduces OR time, because setup is very fast, and the setting of the trajectory alignment is also very, very fast. So I have to conclude with acknowledgements to not only my colleagues from the Department of Neurosurgery for all the different types of robot applications, for depth electrode, for biopsy, Dr. Minchev, who did his PhD thesis on the robot, then of course ISIS, and now later called micro-guided systems, who further developed the robot together with Medtronic, and the inventor, the Austrian Center of Medical Innovation and Technology, especially Gernot Kronreif, who is an excellent engineer, and the support of Medtronic, who made it possible to embark on this very exciting study. Thank you very much for your attention, and I'm open to any questions.
Professor Wolfsberger, we have a couple questions here that we can pass your way. Initially, you had mentioned continuously monitoring, as you're placing the drill guide, drilling, doing your biopsy. How has that impacted just your interpretation of accuracy? I guess adjunct to that would be, what is the learning curve in those regards as well?
Yeah, that's a very interesting question. So again, we are usually not sure what kind of accuracy we have once we log our trajectory. Yeah, we usually set our line with our trajectory at, I don't know, 0.3, 0.4 millimeters. It depends a bit how long we want to take. And then we lock the trajectory and we are blind. Yeah, we know that the system will tell us if we get too inaccurate, but we don't really know what happens. So what we realized is that, first of all, the drilling, stereotactic drilling, must be in axis. If you have a drill like we had, that has a drag on one side, it will create inaccuracy, at least temporary, and it will create continuous inaccuracy, about 0.1, 0.2 millimeters. That's all. So that's why we finally opted for an in-axis drill, like the high-speed drill, like Midas Rex, for example, which doesn't put any or virtually no drag on the whole system and is just more accurate. Okay. So what was the second part of your question, Adam?
Learning curve. If you can speak to learning curve with this system, kind of incorporating it into what was your stereotactic practice prior to this?
So what was interesting is that we tried it out with different surgeons. Some of them had never done a stereotactic biopsy before, yeah, and the learning curve was, as it said, very short, yeah. So it was very easy to acquire the knowledge how to use the robot, and I think the most difficult part of the whole device was the multifunctional arm under the drape. Yeah, this was also the reason why we developed this ratchet type system to lock the arm under the drape, because moving, moving this handlebar of the regular multifunctional arm is very difficult under the drape. And even with the ratchet, we had to teach the surgeons how to do it. But the rest, the robot procedure itself, the instrument, is very straightforward. It virtually doesn't need any explanation. Yeah.
Just reading through some of the other questions. Denny, if you want to jump in as you're kind of sorting through some of these as well, if there's anything specifically.
Yeah, sure. We have some questions here. Let me read one here. So it says, since our accuracy at the biopsy point is going to be more accurate, could that be an argument for a smaller window biopsy needle for small lesions in eloquent areas?
That's a nice question. I think you have to ask this to pathologists. So our pathologists ask us to submit 3 to 4 cylinders of the regular biopsy needle. So they would not be happy if we sample even smaller area. So I think personally the future may be a visual biopsy where we look out of that window, yeah, and see if there is 5-alpha fluorescence or see if there is any spectroscopy even. But I don't think at the moment taking less biopsy material is possible, yeah, because again of all the molecular pathology, all the other diagnostic examinations that the pathologists do.
Thank you. I have another question. What is your preferred registration method for posterior fossa biopsy?
That's a very good question. So, I mean, you all know that registration is generally less accurate with 3D, so skin surface registration, about, let's be honest, 2 to 3 millimeter inaccuracy. And if it comes to prone position, it's even probably less accurate. So it depends very much how large our lesion is. But if we do a brainstem biopsy and the lesion is small, we would of course always do a bone screw registration, because the bone screw registration is accurate to less than a millimeter. So we usually get it to 0.5 millimeters accuracy. So I think you have to really adjust it to the size of your lesion. But what is most important is the robot itself is probably not more accurate than your vertec arm if you do it manually and take a lot of time. Because if you take a lot of time, you can also adjust it to 0.1 millimeter, 0.0 millimeter. Yeah, it's just faster in adjusting it to that accuracy, and it's regular, so it's very consistently adjusting it to this accuracy. Yeah, and so by the end, for your posterior fossa biopsy, I would probably recommend you a skull screw registration.
Kind of along those lines, Professor Wolfsberger, there's been a question regarding stability of the vertec arm. Can you speak to that?
So I would like to give you 2 comments about the vertex arm. First of all, stability, in my opinion, is not a big issue if it's about biopsy needle. A biopsy needle is not heavy, yeah. So doing a biopsy with vertex, I think, is perfectly right. But the problem is that in many cases you're not aligned with your trajectory. So what you do is how the regular vertex arm biopsy works is first you plan your trajectory. Yeah, you plan it on vessels, surface vessels, on deep vessels, on sulci and gyri that you want to avoid. However, then you come to the OR, you transfer your plan. Then you do your biopsy. And then you realize once you did the burr hole, because the burr hole is never done stereotactically, you know that. Yeah, the burr hole is always done freehand, that your burr hole is not perfectly centered. That's very, very common. And then either you take your rongeur and bite and enlarge the burr hole, or you readjust your entry point. And in my experience, it was very common to readjust the entry point. And I think this is not very accurate planning, and it could be even dangerous because there was a reason why we did the first entry point, yeah, and we want to stick to that entry point, yeah. We don't want to readjust it, yeah. So this is the cumbersome part of the vertex, in my opinion, yeah. But otherwise, from stability, I think that's not an issue.
In those regards, the ability to, if you sense that you have gone off of your trajectory, the ability to realign, how often did you find yourself doing that with a precision aiming device versus the ability to quickly realign with the AutoGuide? Additionally, can you speak to skull fixation, so you have a second point of fixation with the drill guide?
So I think it was more the rule than the exception that we realigned our entry point. Yeah, that was nearly always because we wanted the entry point in the center of our burr hole, and the burr hole was never centered. Yeah, and in terms of the skull second point fixation. So I think the idea of fixating the robot with the guidance tube to the bone is very interesting because it creates a circle. So you have the Mayfield clamp. From the Mayfield clamp, which fixates your skull, your head, you have connection to the robot, to the manipulator of the robot, and again to the skull via the tube. So this circle is very, very stable. Yeah, and I think this is the reason why this minimally invasive instrument set will finally be available for DBS, for example, and for the Visualase system, because this circle and this double fixation, this 2-point fixation, maintains stability, which vertex, of course, cannot do because there's always elasticity between the arm and the skull.
Thank you. An additional question: have you found any approaches to be prohibitive with Stealth Autoguide?
Mm-hmm.
I will add to that one because it's related. So, can biopsies be done everywhere in the brain, even in the cerebellum? So I think it's related to this other question.
Yeah, that's a good one. So we found out that the most difficult procedures to reach the whole skull are, of course, the depth electrode procedures, the bitemporal depth electrode procedures. Yeah, and this is why we developed a centered attachment where the robot arm is actually fixated in the axis of the Mayfield screw, so in the center. So you can reach by both sides equally. So there's no difference if you move the robot to the right or to the left temporal area. So this is for this area. So I think you can reach all parts of the skull with one positioning. Maybe not front and posterior fossa at the same procedure, but honestly, I've never heard of a procedure that necessitates this. And to your second question about the cerebellum, yes, of course, in prone position, that's no problem at all to reach cerebellum. Side position, no problem. Yeah, we've done a lot of them. Okay.
Just going to go back to learning curve. For you to teach, a resident fellow, another colleague, can you speak a little bit more towards the learning curve and kind of adapting to that? I remember at one point you had stated that this is integrating robotics into a workflow that you already know, whether that be navigation or using the Vertec arm for precision device, but learning curve, going back to the learning curve.
Okay, so I mean you can consider the AutoGuide robot as a different attachment to your multifunctional arm. On the one side you have the Vertec positioning system, and on the other side you have the AutoGuide. So different heads of the same arm. Yeah. And if you know how to do a Vertec biopsy, it will be much easier to do the same biopsy with the robot, because that's what it was designed for. And, you don't have to do all the cumbersome adjustments of the precision aiming device. The thing that you need to learn additionally, if you want to use the minimally invasive instrument set, of course, is yes, how to lock these to the bone, how to do a step incision, to the ideal size of your skin incision to be able to introduce the dilator without any drag of the galeal flap. Yeah, because of course that's what we initially realized. When your skin incision is too small and your galeal flap is very thick, like in the posterior fossa, and then you enter your guidance tube, it will be there will be drag put on the guidance tube, and maybe accuracy will diminish. But this is why we always used the K-wire, like a needle, to lock it centrally to the bone, and then advance the guidance tube around it. So this is something I think that has a learning curve, that needs some experience, but again, it's still a very simple procedure for a neurosurgeon, I think.
Hey, doctor, we have another question here. So it says, how is clinical important, other than cosmetic, is reducing the incision size from 14 millimeters to 3 millimeters?
That's a very good question. Yeah, it's probably not. It's probably not at all. But I remember doing temporopolar biopsies and doing a biopsy just behind the eye that is like, I don't know, 3 centimeters deep through the temporal muscle. That's hard. Yeah, and if you have done it, you know what I'm talking about. And then we're not talking about 14 millimeter. We're talking about a large incision if you really want to divide the muscle and see the bone. Yeah, and do a burr hole, a real burr hole there. Yeah. And the same is true for some posterior fossa biopsies. If you're deep enough, the muscle can be really thick. Yeah. So, and as I showed the picture, this was a real surprise for us, that now the skin incision is so small, even if the muscle layer is very, very big, because you don't have to free the bone. You don't have to see it. Yeah. You're drilling through that small tube, no matter if your layers above are thick or thin. But of course, you're completely right. If you need a biopsy for a malignant glioma, it's probably no difference if it's 14 or 12 millimeter.
Thank you, doctor. Another question here. How will you convince your other neurosurgery friends to use the robot if they have been successful with the Vertec biopsy for years?
It's also a very good question, because honestly, even in my own department, I couldn't convince some of the people that were successful with Vertec arm for years and that considered the robot just an expensive tool for the exchange for Vertec arm. Well, if you have used it, then you also know how difficult it is, to adjust it to 0.0 or 0.1 millimeters. Yeah. So, and if you know that and you see the robot once how fast this works, then you should be convinced if you're somehow technologically orientated. Because that was the same for me when I saw the robot the first time. I did many, many vertex biopsies before, and I thought, oh, why do I need this? But on the other hand, I knew how cumbersome it can be to achieve accuracy and how impossible it can be sometimes. Yeah, and with a robot, it's just there. It's just inherent in this system. And I think, especially different applications, future applications like the endoscopy holder, they make sense to use a small robot device that you can adjust very, very accurately. Yeah, so I think you shouldn't limit it to the biopsy. You should think broader for depth electrodes, fast positioning, and very precise positioning for future applications.
Thank you, Doctor. This is another clinical question. For brainstem lesions, did you always perform frontal boreholes?
No, not at all. Honestly, we never did, because the biopsy needle is not long enough for most brainstem, for deeper brainstem lesions, like pontine lesions. The biopsy needle, in many cases, is not long enough for a frontal approach. So we usually used a transcerebellar approach for the brainstem lesions. Yeah.
Thank you. We have another one. So currently there are more invasive ways of doing biopsies, like stereotactic cases. Do you see the robot replacing these procedures?
I'm not sure if I understand the question.
Yeah, it says, are we just focusing on stereotactic cases? Currently, there are more invasive ways to do it. Do you see the robot replacing stereotactic biopsy?
Well, I think the robot is just another means of a stereotactic biopsy. Yeah. I mean, usually, if we're talking about more invasive biopsy, that's an open biopsy, isn't it? But usually, there's a reason for open biopsy, like a surface lesion, yeah, or a very dangerously located lesion, maybe even very eloquent located lesion where you need some stimulation, intraoperative monitoring. No, I think the robot can replace your frameless stereotactic system that you have, yeah, and make it probably faster and or even more accurate, constantly high accuracy, yeah.
Yeah, so yeah, they were talking about open biopsies.
No, I think there's a reason for open biopsies. No, usually, I don't think the robot can replace it. Yeah.
Okay, thank you. I don't know, Adam or any of the panelists have any other questions? We have a lot of thank you messages, Dr. Wolfsberger. I just want to let you know. Great feedback from the participants as well.
Great, thanks a lot.
Well, there's one in here that you mentioned that it's more accurate to use bone fiducials. So they are asking if you have measured the accuracy of bone fiducials versus tracer registration. I think you have a paper on that.
We have never measured or assessed the difference in real accuracy, so real target accuracy on post-operative imaging between bone fiducials and surface merge. But we know that from registration accuracy values that you get, you are usually about 0.5 millimeter accurate with bone fiducials, and you about 2 to 3 millimeter accurate with skin surface registration. And of course, this gets even less accurate with prone position. Yeah, I think we know that. So bone fiducials, yeah, they are accurate to the system inaccuracy. So less than 1 millimeter usually. I think that what is important to know with bone fiducials is that you have to find the right bone fiducials. Yeah. So it took us a long time to find, let's say, easy to use bone screws Yeah, and the ones that we found now, and they are in one of the last publications mentioned, you can screw through the skin, yeah, and theoretically you can leave them inside even overnight, yeah. So I think they are just perfect. They're self-cutting through the skin into the bone. Yeah, they don't bleed or anything. They are just ideal. Yeah, and they have the right head, screw head, to for the pointer to enter.
Denny, if you want to, let's see a couple more questions coming through. If we want to leave off with one last question, and then we are at the top of the hour, we'll go ahead and conclude our discussion for today. Was there anyone that was specifically standing out?
I'm reviewing the questions. I think, well,
we've got one here at the bottom. What do you see preventing the use, the Stealth Autoguide use with or for DBS?
It's always the hardest question at the end. So don't forget with DBS, every functional surgeon has his own workflow. And this own workflow is perfect. Yeah, it has been perfectioned to the 0.1 millimeter inaccuracy on post-op imaging. And you have to cope with it. Yeah. And honestly, I believe that most of these inaccuracies are better than an optic navigation system can deliver. So even the camera bar, a movement of the camera bar, a movement of the pointer generates higher inaccuracy than a DBS procedure can take. So first of all, we have to rely on a different technology than optic navigation. As I said, we have to rely on the robot's internal positioning capability, which is less than a tenth of a millimeter, probably hundredths of a millimeter or less. You have to ask our engineers for that. But it will be highly accurate enough to position the electrode so that you can compete with a leg cell frame. Yeah, and we believe that in the end, if we develop such a system that rigidly mounts the robot to the head, to the patient's head, the position is probably at least as accurate as a leg cell frame. But this is what has to be done, this mounting the robot rigidly to the head. So you need a very, very solid frame or head clamp, yeah, nothing like a Mayfield. A Mayfield is inaccurate by itself. It moves between its two pieces, yeah. And then you have to mount the robot to it. So I think we need some more time, but in the end, we'll have something, but that is not relying on optic navigation, yeah, just to let you understand. Mm-hmm.
Great. Well, I think with that, let's go ahead and close. I want to thank everyone for joining, and you know, more specifically, Professor Wolfsberger, for sharing your experience. Thank you for your continued collaboration. I also want to note, please join us for subsequent webinar calls. The next one being June 9th. It's going to be at 7:30 Mountain Time with Dr. David Allison, who was the first in the U.S. To use the Stealth Autoguide for sEEG. He's additionally using it for Visualase. Additionally, there was an attached document that will allow you to register for these subsequent events. And with that, I thank you, and I hope you have a wonderful day.
Thanks for listening. Thank you, Adam, for the invitation. Goodbye.
Thank you, everyone, for joining today's event. You may now disconnect and have a great day.